Well, a very warm welcome to this event for our participants that focuses on our imaging study, where we aim to inform as many of you as possible about our fantastic imaging study, what it involves, why we want to do it, and to give you some examples of the type of novel and innovative research these data are already enabling. So it's now 20 years since UK Biobank was first imagined as being a resource with the potential to benefit millions of people in the UK and elsewhere. And thanks to the commitment of all 500,000 UK Biobank participants, we're now arguably the world's most significant resource for health-related research, using the data from the questions you answered at recruitment, the physical measures and biological samples that were taken, as well as following up your health over time through linkage to your health records. So UK Biobank's imaging study started in 2014 with the aim of inviting up to 100,000 participants to attend an imaging assessment clinic to obtain data from MRI scans of different parts of the body. And we're now just over halfway through. The study is the largest in the world by some margin to perform a range of MRI scans for research purposes. And these data, as we'll see in this event, are already revealing new insights about disease mechanisms. So first, some practical tips to help you get the most out of the event. So there are several engagement tools at the bottom of your screen. One of the most important is that you can enlarge each of the screens just by clicking on the small square in the top right-hand corner. Please feel free to ask any of your questions through the Q&A thumbnail that's at the bottom right of your screen. And where possible, I'll try to address those questions to the panel at the end of the event, which will last about an hour. If not already doing so, we recommend to use Google Chrome for this event. And also to let you know that we hope to make this event online for those who can't watch it in real time in the next day or so, and there will also be captions available. Okay, so on to our three fantastic panel of speakers for today. We have Mr. Steve Garrett, who's the UK Biobank's Imaging Programme Manager, who's been leading this study since the start. Professor Paul Matthews, who's head of the Department of Brain Sciences at Imperial College London and who leads UK Biobank's expert working group on imaging, and Dr. Anya Tapiwala, who's senior clinical researcher and consultant psychiatrist at the University of Oxford, who's done some fantastic research using the MRI scans. So without further ado, I'd like to introduce Steve Garrett, who's the Programme Manager here at UK Biobank for our imaging study, and we'll talk about the background and the development of the study. Over to you, Steve.
Thank you, Naomi. So yes, as Naomi says, I'm Steve Garrett. I'm the Programme Manager with UK Biobank, and I've been working on the imaging enhancement study basically since its funding was approved, which was back in 2012. So the imaging enhancement study itself is exactly that. It's an enhancement to our original baseline study. So we had our 500,000 participants take part in that baseline, and when that completed in 2010, our scientific community recommended to our funders that we look at doing this imaging enhancement. We went through a whole application process and worked very hard to narrow down exactly what would go into that imaging study. And eventually that was approved, as I said, in 2012. And that was to do 100,000 of our participants, so roughly 20%. And they would undergo an MRI of the brain, the heart and the abdomen, plus what's known as a DEXA scan, so it's whole body bone density, and an ultrasound scan of the carotid arteries. And as part of that imaging visit, we're also doing a repeat of some of the baseline measures that were conducted in the original study. Now, when we received that funding, it was initially for a pilot of approximately 5,000 participants, and we needed to show that that would work and that we could get high quality images from the required number of participants. And then when that was proved to be successful, the remaining funding was released. So just a very brief look at the timeline for that. So as I said, the funding was agreed in late 2012, and then we focused on finding a location and sourcing the scanners and the staff and building the sites. And the pilot study began at our sites just outside of Stockport in April 2014. And that pilot study ran for a couple of years. And the funders came back to us in early 2016 to say that they considered it a success, and that released the remaining funding. We then searched for additional sites, and the first one that we opened outside of the pilot site in Stockport was the one that we have in Newcastle, and that opened in April 2017, coming up to 5 years ago now. We also received some funding to what's known as the Dementia Platform UK study, which was a small number of repeat images, and that commenced in 2018, 2019. And we opened another site in June 18 in Reading. Then our fourth and final site opened in Bristol in February 2020, which, as I'm sure many of you realise, was probably the worst possible time we could have opened a new site. Because soon after, in March, we actually closed down the imaging centres for some 12 months. Whilst the pandemic took its toll. But actually, we reopened the sites just over a year ago to conduct a COVID study on our own right. So we had a couple of thousand participants, half of whom had COVID and half of whom didn't. And we did a case control comparison study of before and after images, which is pretty unique in the world, and some of you may have seen that was actually some results from that were published a couple of weeks ago, and it was picked up by national news. And where we are right now, we've just had funding approved to do a full cohort repeat, whereby we're going to invite all of the 100,000 participants from the baseline imaging back to do a further study. So I'll just talk a little bit about the visit itself. So you arrive at the site, and you are welcomed, and you undergo a screening. So what that basically means is we'll ask you a few questions about your health and medical history, just to make sure that it's safe for you to go into the MRI scanners. So a very obvious example: if you have a pacemaker, then it's not safe to go into the magnets, and so you wouldn't be allowed. And I should say, we do this screening before you arrive at the site via a phone call when you book your appointment. You then have the first of the scans, which, again, depending exactly on how the visit is running, will be a brain MRI, and that lasts for around 40 minutes. And I should say at this point, the scanners, they can be noisy, and they make some very unusual sounds. So I'm going to attempt to play some to you. I'm not quite sure how well this will come across, but here are some noises that you might hear in the MRI scanner. I'm hoping you can hear these. It sounds very much like alarms often, or hammering noises. And I know that these can be a little disconcerting. But they are completely normal. So whilst you're on the scanner itself for about half an hour, you've all of these strange noises going on in the background. But it's completely normal, and it produces these really high quality images. So once you've done the brain scan, you'll then go on to have on a separate scanner, a heart and whole body MRI scan. And then in separate rooms again, you will have the DEXA and then the carotid ultrasound. And the idea is that 3 participants will arrive at the same time. 1 will go into the brain scanner, 1 to go into the heart and abdominal scanner, and 1 for the DEXA and the ultrasound, and then everybody swaps over. Once you've had the imaging, as I say, we'll do a repeat of the measures that were done at baseline. So we will collect blood samples and the urine sample again. The repeat of baseline is physical measures, the study interview, we do the touchscreen questionnaire, and there are some cognitive function tests as well that we perform. And then you'll be free to depart the site. And that entire visit lasts around 4.5 hours. So it's quite a long visit. We appreciate that is an awful lot to ask, but we get some incredibly valuable data from that. So where we are currently, I think Naomi mentioned before, we're about halfway through the baseline imaging visit with just over 50,000 participants. And more than 13,000 of those have been scanned at our Newcastle site. We've done around 3,000 of the dementia platform repeat scans and a couple of thousand repeat visits for the COVID study. So from those 55,000 appointments, we've had some fantastic feedback. So we will ask you to give us your thoughts once you've visited the site. And from there, around 95% of people who fill out that form describe their visit as good or excellent. 85% or more of those participants say that they would be happy to return again within a year. And 99% have said that they felt safe, which, again, in these present times during the COVID study, that's really important. And we do have various secure measures in place at the moment, and they will remain in place until it's safe for those to be stood down. So staff will be wearing masks. We will physically distance wherever possible, and we have an enhanced cleaning regime in place. In terms of the location, so the site is just outside the main city centre in Newcastle, just off the quayside there. So this is the Tyne Bridge, the yellow line going over the River Tyne there. That's the Tyne Bridge. So it's about a mile from the centre. And it's actually situated in what looks like a warehouse, because it was a warehouse. But we've converted that into what looks like a state-of-the-art centre. We have a large area where you'll do your touchscreen questionnaires and where you'll be welcomed to the site. And this is one of our scanners, and this is the view that you would get as a radiographer looking through the observation window there. And you can see it's actually quite a wide bore scanner. It's a larger MRI scanner, and it's got a lot more space within it. But it's a really nice location. So what about the future? Well, we're going to hopefully complete the baseline imaging in the next couple of years, by the end of 2023, maybe into 2024. We're going to start doing the full cohort repeat imaging in 2023. So that will begin actually in Newcastle. So we're really pushing to try and get as many people through their baseline image in Newcastle as possible before we start on the repeat. And then who knows what else? There may be further imaging, maybe further COVID imaging, and maybe a third repeat, but lots more to come. And I'll very quickly, I think I'm just coming up to my time, but this is the sort of research that you're able to do. So these are typical MRI images that we get from the abdominal scans. And these show 2 gentlemen of a similar age a similar body mass index and a similar percentage of body fat. But actually, when you examine the MRI scans, you get very different amounts of internal fat. So the person on the left-hand side has nearly 6 litres of internal fat, and on the right, only just over 1.5. And this is the only way to see those sorts of measures. And I think one of our researchers is going to talk a little bit more about the brain MRI in a little bit. But as I said, very recently, there is a paper being published in Nature that was picked up talking about the COVID study and the impact on the brain. And that was only possible via the UK Biobank imaging study. It's the only place in the entire world where that sort of research was possible. Okay, I shall stop now, and I think, is it Paul? Are you taking over next?
Yeah, I'll introduce Paul. Thank you very much, Steve. So I'm delighted to introduce Professor Paul Matthews, Professor of Translational Neuroscience and Therapeutics, Head of the Department of Brain Sciences and Associate Director of the Data Science Institute at Imperial College London. If that wasn't enough, he's also Chair of UK Biobank's Imaging Expert Working Group and has been absolutely critical in getting this study established and for making the scientific case to have such large-scale data. So he'll talk to us today about the value of imaging data for health research. Over to you, Paul.
Great, thanks, Nye. And let me say to all of you who are listening, it's a real privilege to be with you today, partly because it's been such a privilege to be part of what UK Biobank is doing. UK Biobank is enabling researchers across the world to answer questions that affect each one of us. So I know that I asked, and I suspect you do too, what diseases am I at risk of suffering from? How can I minimize the risk of disease as I get older? Of course, we all want to know how diseases can be diagnosed before they're too advanced to be treated. And we want to help identify new and better treatment, which we all agree must be needed, whatever the disease. What you and others are doing in UK Biobank is helping researchers find the answers to all of these questions. And what's so extraordinary about it is researchers all over the world picked up the challenge and are working on it. Let me tell you a little bit specifically about what is being done with some of the data from the imaging enhancement that Steve told you about, which has really changed the game for what UK Biobank can be used for. Now, Steve showed you two people and highlighted their differences. I think the body images are always a great place to start because every one of us watches our bodies as they change over life. And what you see here are a group of UK Biobank participants, just like you, who've been imaged. But what we see here is just a slice of the images through their body. And what it highlights are the differences in tissues that have been labeled with these artificial colors that we see. So here, for example, these bright colors in this image are muscle in the thighs, as you can see. This blue here on the edge of the third image is fat just beneath the layer of the skin. This red down here is the fat around the body organs. And what you can see is all of us vary enormously with regard to the distribution of fat. Some people have more fat under their skin and in their body. Some people have relatively little fat within their organs, but more around the outside of the body. And we're beginning to see that this is really critical. Now, why is this so important to understand the distribution of body fat alone, for example? Well, work in UK Biobank, led by Jimmy Bell at Westminster University and his colleagues, has really shown us how we can take a big step forward in our understanding of risk for individual people of suffering from big diseases of mid and late life, coronary heart disease, type 2 diabetes, and course, the metabolic changes that lead to that. So what they were able to do is look at the distribution of body fat, fat in the subcutaneous space, fat around the organs, fatty changes in the liver, and fatty changes in the muscle, and develop a curious, visually really attractive index, the STAR index. It tells us, helps us identify people at very low risk of any of these diseases, and people, alternatively, who have higher levels along each of these different axes of liver fat, relative muscle fat to muscle ratio, relative abdominal fat. Those people who have a lot of fat that's distributed in organs as well as around the organs in the body and subcutaneously have very high risks of all of these diseases. One can quantify, you can't read the numbers here, they're not terribly important, but you can quantitatively measure the distribution of body fats and find which fat distribution patterns are associated with which risks of disease. For example, fat in the skeletal muscle, the big muscles of the thigh, is a strong predictor of risk of coronary heart disease. Let me just summarize by telling you why this is so important. Lots of epidemiology looks at the relationship between various factors in the population and risks of disease. It's hard to pick up the newspaper without finding an example of this. But what's special about the imaging is by being able to look within organs of individuals, it's taking this prediction to the new level and allowing us to do the precision medicine of identifying risk by person. Let me move you on to another study that it's about how we can minimize risks of disease as a population at large. And this is about the things we do, the exposures we have. This is what's really special about Biobank. The information you've contributed on where you live, the diet you have, the activity levels that you partake in each week is really beginning to shift the way we can analyze the risks of disease with respect to exposures in population that are common to all of us. One of the most important of these is alcohol. Most of us enjoy a glass of wine now and then. I must admit that it's a sobering, Biobank has helped us make a sobering reflection on what the potential impact of alcohol is at a population level. By looking into the UK Biobank data set and being able to image the relationship between changes in organ systems, the brain, the heart, the liver, with differences in alcohol consumption across the UK population that is represented in UK Biobank, we were able to discover some rather sobering facts. First of all, we found that even moderate alcohol intake, 1 to 2 glasses of wine, is associated with reduced brain volume. Here you see on this artificial color image an expression of the areas of brain in the population that showed significant reductions in the volume, indicating degeneration with increased alcohol intake, even at moderate levels. We found evidence for increased heart size. Increased heart size is bad when you're in midlife. Reduced flexibility of the large vessels, a sign of a higher risk of atheroma, and increased liver fat. Doubling alcohol consumption, even going from 1 to 2 glasses of wine per day, was associated with loss of brain volume in the areas illustrated here, equivalent to half a year of aging. This really provokes a rethinking of what government policy is in this area. And it helps us all understand that what we do in everyday life has an impact on our long-term health outcomes. Well, the next thing I want to talk about is how diseases can be diagnosed between the before they're too advanced to treat. And I want to talk about a very special study that Steve Garrett mentioned earlier, and that was the UK Biobank longitudinal study of the effects of exposure to SARS-CoV-2, which is the virus that causes COVID, I'm sure you all know by now, on the brain, heart, and body. The group of people who had volunteered to take part in the imaging study before the pandemic, almost 50,000 of them, provided a really unique resource because these people had been imaged within a short period of time before the pandemic started. And so if they were re-imaged after they had been exposed to SARS-CoV-2 in the environment, either with development of COVID, symptomatically or not, we could understand what the effects were by looking at the changes in brain, heart, or body size, shape, and characteristics by imaging before versus afterwards. It really was unique. The ability to look over time longitudinally with these incredibly precise imaging measures that allow us to look at areas almost too small for most of us to pick out with our eyes, we can begin to find evidence of very early changes that may have major consequences later in life. Let me give you an example of this, which is shown right here. This is from a paper that was published just a couple of weeks ago in a major journal and carried in the news media, not just nationally, but across the globe, on the front page of the New York Times. And here you see, this is a sort of what we call a blown up brain. It's taking the brain and blowing it up like a balloon. And you see the wrinkles in the brain highlighted as different shades of gray. And here in the bright orange red, you see regions of brain that showed shrinkage after exposure to SARS-CoV-2 relative to before. This is really remarkable because this is the first study in the world that was able to look at people before and after this exposure. Now, is this of any importance at all? Well, we think it might be, because here, this looks at the results of testing cognition in people. And this testing of cognition for those who participated in the imaging study was done at baseline, and then exactly the same way after exposure or not to SARS-CoV-2. And what one can see is that in those people who weren't exposed to the virus, they showed some small changes in performance on this test, which involves making a line go from circles, from one circle to the next, with sequentially increasing numbers. But in people who were exposed to the virus, it took them longer after exposure to the virus than before. And you can see this is beginning to be significant. This was even more significant because if you looked at a more difficult version of the same task in which people had to make a line connecting first from 1 to the letter A, and then to the number 2, and then to the letter B, and so on, to 27 circles. It's takes a bit more thought because you've got to keep shifting back and forth between numbers and letters. It took them even longer after exposure to the virus. So we fear that this small amount of brain volume loss, just 1% or 2% of the total brain, actually reflects changes that are more widespread. It's urgent that we follow this up now and understand whether this is going to reverse in time or whether there is a risk that it might be a harbinger of changes to come. Well, Biobank is allowing us to study all of the organs, and I wanted to highlight 2 simple points in closing. And the first is that one of the keys to finding new and better treatments is measuring things more precisely. And UK Biobank has, by providing the biggest imaging set to researchers available anywhere in the world, has catalyzed the use of a whole range of new methods in artificial intelligence and others in order to measure size and shape accurately. This is beginning to actually change the way radiologists work. because they don't have to now measure things by hand, but it can actually begin to use these sophisticated pieces of software. Up on the top is what's called a neural net, or at least a pictorial description of a neural net that was used to create precise measurements of the heart. These are all different regions of the heart. And I want to point out particularly from the MRI of the chest of a biobank imaging enhancement participant, how one can pull out the ascending aorta and the descending aorta, the two great sections of the vessel leading from the heart. Now, one can use these measurements, which now, given the algorithm, one can perform at great scale, so that 50,000 people who've been imaged could all have the images precisely defined in size and shape. One can relate these to other information in UK Biobank, one of the most important of which is the genetic data that has been provided, as all of you have contributed cells that can be used to harvest the DNA. This allows us to do what's called a genome-wide association study, looking for the genes that are responsible for the trait. And what we've discovered in looking at the ascending aorta is that the flexibility of the aorta, the distensibility, the way it changes size with each heartbeat, is related to a particular molecule called transforming growth factor beta. What's important about that is the higher the expression of transforming growth factor beta, it appears the stiffer the aorta. Why is that important? Well, large vessel stiffness is a big risk factor for atherosclerotic disease. And it's posed the real question of whether we can begin to use a new generation of TGF-beta inhibitors being developed for cancer to reduce the risks of late-life cardiac disease. So let me bring us to the close. I shared you at the beginning that participation in the UK Biobank imaging enhancement, like the rest of UK Biobank, is helping us answer some really important questions, not just for the population, but for each one of us. It's also, I'm sure you appreciate, you have the privilege of participating in a study that is beginning to help others. And the more that one is able to do for it, the more one is able to help researchers develop the tools, the ideas, and ultimately kinds of treatment changes that will help others. So let me close with my huge personal thanks and also acknowledging all of the tremendous work that the UK Biobank Imaging Working Group did in setting this up, but also, or in guiding the setup of this, but also the UK Biobank Executive and the Imaging Center teams led by Steve for making it all happen. And finally, of course, most important, underlying all of this, is all of the UK Biobank participants who've so generously given their time and energy to support the building of this absolutely unique imaging resource. Thank you.
Thank you very much, Paul. So our final speaker today, who I'm delighted to introduce to you, is Dr. Anya Tapiwala, who is a senior clinical researcher and consultant psychiatrist at the University of Oxford. And a couple of years ago, she was awarded a prestigious Wellcome Trust Clinical Research Development Fellowship Award to specifically study the effect of alcohol consumption on brain health, which she'll talk to us about today. So over to you, Anya.
Hello, thank you very much for this invitation to speak to you all. So I'll start with the problem, and this is it. Drinking of alcohol is endemic across the world. And whilst we know that very heavy drinking over long periods of time can really harm the brain, causing shrinkage and even dementia, the story for more moderate amounts has been quite contentious, with some claims that it could be protective for the brain. And that's really led to what we call the J-shaped curve. And the theory is that, yes, okay, heavy amounts of alcohol can harm your brain. Here I'm showing the case for the brain, but the same argument has been made for the heart. But actually, perhaps a glass of red wine a day might be good for our brains, better than drinking nothing. But the findings in moderate drinkers with looking at brain scans have been quite conflicted. So while some studies have shown beneficial effects or apparent beneficial effects, others have shown associations with harm. And there are many reasons, I think, for this, and one of which is that most of the studies before UK Biobank were quite small, limited number of people. Perhaps people haven't accounted for lots of other factors that can be important when thinking about brain health. So really, the UK Biobank imaging sample has kind of changed the game for researching these kinds of questions. With the huge sample size that's already been mentioned today, we can really examine for very small effects of alcohol on the brain. We can distinguish between people that have never drunk alcohol and people who used to drink, and that's quite important when thinking about this question, because people who used to drink may have stopped because of various health problems. And that could lead to perhaps spurious protective associations of moderate drinking. We can account for all these host of other factors that could contribute to brain health. And finally, because we have imaging in not just the brain, but also liver and other organs, as you've already seen today, we can start to look at crosstalk between these organs, which is very important when thinking or trying to understand mechanisms. So for a quick crash course on brain anatomy, because I think it will help you understand some of the results I'll be showing. If you imagine your brain like this, and we make a cut along this black line and open up the two halves, inside you'll see the brain, your brain looks like this. And you can see two different types of brain tissue, it's different colors. So around the outside is what we call the gray matter. And then deeper within the brain, we have this white matter, because the brain cells in here are sort of insulated, if you like. If you think about an electrical cable with the wire in the middle being the brain cell and that insulation layer around the outside, the same sort of analogy can be used for the brain. And with the brain scans that we have, we can start to assess these two different types of brain tissue. So what we did first was we looked at how much people drank in terms of units per week, and the relationship between that and their grey matter, so that outer layer of brain tissue that I just showed you. And we found the more people drank, the less of that grey matter there was. And here you're looking at a kind of summary image of more than 25,000 people's brain scans, and all the areas in blue here are where we found that relationship. So more alcohol, less brain. And this is taking account of lots of other factors that may affect your brain health. So how old you are, your sex, your blood pressure, your BMI, medications you're on, smoking. We then looked at the relationship between alcohol and that inner layer of white matter, so that those insulated fibers inside the brain, if you like. And again, we found the more people drank, the lower quality of that insulation. So here you're looking at all these blue blobs are where we saw that relationship. More alcohol, lower quality insulation, if you like. And the question, of course, everyone asks is, okay, well, alcohol's bad, but how much is too much? And we wanted to drill down. And so we compared people in terms of how much they drank. So we compared people that drank less than 7 units of alcohol a week. And that's indicated by this dotted line here. And then people that were drinking more. So I've got men on the top here and women down here. So anything to the left of this line means smaller brain compared to those drinking at less than 7 units. So hopefully you can appreciate that in men and women, anything above 7 units of alcohol a week, we started to see smaller brains. If you want a reminder of what 7 units a week of alcohol looks like, perhaps you'll think it's not too much. So it's either 2 large glasses of wine or 2.5 pints of beer. And this is per week. But before you all panic and pour your bottles of wine down the sink, the effect was widespread across the brain, but it's quite small. So if you look at this left-hand pie chart here, of the things we know that affects how large your brain is, the green slice, the small green slice here are the things we can do something about. We call them modifiable factors. So this big blue section, things like age or genetics, we can't change this. However, if we blow up this green slither into a pie chart of its own, of the things we can do something about, alcohol in orange here actually represents quite a large chunk. So it seems to have more impact than other things that we could change, like smoking or BMI or our blood pressure. We also want to look at, well, does it matter when you drink? So if you spread your drinking through the week, rather than saving it up to have it in one sitting. So we looked at the frequency by which people binged, and by that I mean, well, that was defined as more than 6 units of alcohol in one sitting. And we found some evidence that if you did drink all your alcohol in fewer sittings, that might be worse for your brain than not binging. And in contrast, what you drink seemed to matter less. So we compared people that solely drank wine compared to solely drank beer compared to solely drinking spirits. And actually, we couldn't really see a difference, at least in terms of the size of people's brains. So it seems to matter how much you drink in terms of units of alcohol per week, not what you drink it in. However, if you have higher blood pressure or higher BMI, the effects of alcohol on the brain might be exacerbated to some degree. So just looking at the bottom graph here, for instance, as you drink, or if you drink more, you get a smaller brain. So grey matter volume is literally the volume, the size of your brain. That decrease with alcohol is steeper if you have a BMI. So the solid line here is showing you if your BMI is 40, that reduction in brain size when you drink is much steeper than if your BMI is 20 in this pale dotted line here. And the same goes for blood pressure. And just finally, I'll mention another body of work we've been doing, really making use of this multiple organ scanning, which we have in the biobank, which again is kind of completely novel. The level of iron in your brain increases as we age, and it has been, higher levels of iron have been associated with things like Alzheimer's disease and Parkinson's disease. And what we've seen so far in biobank is that people that drink more have more iron in their liver and brain, which we can detect on a certain type of imaging. And that in turn seems to associate with faster decline on some of the memory test scores. So I think this is a kind of exciting avenue which we're kind of pursuing and really makes use of that sort of multi-organ imaging. So just to sum up, what we've found so far in this huge, very valuable resource is that even small amounts of alcohol seem to associate with smaller brain volumes, lower quality insulation of brain tissues, and higher levels of iron in the brain and liver. And I think it's really valuable to think about our findings in the context of kind of current international alcohol guidelines. So if you remember, we started to see these harmful associations at just 7 units of alcohol a week and greater, which is double what our current low-risk drinking guidelines are in the UK. And actually, we're pretty conservative when you look internationally. So I think it will be interesting to see if these are revised downwards in result of ours and others' findings in the biobank. And of course, with the advent of the longitudinal imaging, that's going to be really important because everything I've shown you is on one brain scan. And what will be really persuasive is if we can say, well, this is what's happening in people's brains over time if they drink more. So having that second brain imaging at a later period of time is going to really take this research to the next level, I think. So just a final word of thanks to, of course, all of you, of which we couldn't do this work, and the funders and my many collaborators in the UK and elsewhere. Without this, work couldn't be done. Thank you.
Thank you very much, Anya. So I'd like to move on to the panel discussion now. And I wondered, first of all, if I could just introduce Professor Sir Rory Collins, who is Principal Investigator and CEO of UK Biobank since its inception in 2005. And I wondered, first of all, Rory, perhaps if you could give a broad overview for the participants about what in particular do you think makes UK Biobank unique?
Yes, thank you. Can you hear me, Naomi?
Yeah, fine.
I think the thing that there are 4 dimensions that I think, when I think about UK Biobank. The first is the scale, obviously, with 500,000 people. There are very few studies that have been able to recruit such large numbers, and I think that's a testament to the altruism of the UK population. The second is the depth. We've seen that in the presentations today in terms of detailed imaging. It's worth bearing in mind that prior to UK Biobank, the biggest imaging studies were only a few thousand people. And in fact, when we first had discussions through Paul Matthews with the imaging community around the UK and internationally, when we said that we were interested in doing an imaging study of 100,000 people, they thought it was a typo in our email invitation. They thought we meant 10,000. So having such large scale and such depth with the imaging, but also so much other information about lifestyle, environment in which people live, and increasingly now assays of the samples, biochemistry measurements like cholesterol and markers of diabetes status, sequencing data, whole genome sequencing data on 500,000 people. And again, bear in mind that the first person sequenced was only 20 years ago. So again, on a scale which is unprecedented and depth. I think now duration, so the recruitment in 2010 to 16, 6 to 10, means that we have about 15 years of follow-up of health outcomes in these, all the participants, the people who are on the line, and all of your colleagues. And I must say, myself, I am also a participant. And then finally, the 4th dimension, and the one that's really transforming health research around the world, is access. The resource, the de-identified information about all of the participants, all of this scale, depth, duration is available to researchers around the world to do all kinds of health-related research. You've seen some very nice examples from both Paul and Anya. And there are some 30,000 researchers around the world using the UK Biobank data to study all sorts of health outcomes. And the combination of those 4 dimensions really sets UK Biobank apart from anything that exists anywhere in the world, which is why so many researchers outside of the UK are using it, because it's so fundamental to understanding biology.
Great, thank you very much. Moving more specifically now onto the imaging study, Rory, I wondered if you could perhaps tell our participants a bit about whether any results will be fed back to the participants from their scans, and under what circumstances we do that. I mean, I know we've discussed this quite a bit at our end.
Yeah, it's a very, on the face of it, it seems very obvious that if anything was seen on a scan at any time, one would feed it back. And this was one of the initial discussions we had with the funders. And so as part of the piloting study that Steve described, we did a very careful comparison of the effect of looking at the first 1,000 scans in great detail, having expert radiologists on brain imaging, on heart imaging, body imaging, to check all of the scans to see if they could see anything that they thought was unusual. We then compared that with the approach that we'd taken in the baseline assessment visit for UK Biobank, which was if something was noticed during the visit that raised concern, then that would be checked, and those incidental findings could be reported to participants and indeed to their doctors. So we kind of compared this sort of more incidental approach, where if the technician doing the imaging scan noticed something during the scan, then that would be brought to the attention of the team. Radiologists would look at it, and if they thought it was a concern, then they would report it to the participant and to their doctor for further investigation, compared with a much more systematic screening of the images. Now, on face value, you might think the latter, systematic screening, would be a good thing. But in fact, what we found was that we picked up a lot of what could be called false positives. So we picked up lots of unusual observations on the imaging, which were then investigated. Sometimes those investigations were quite invasive, and they turned out not to be important, to be just kind of normal anomalies, if you like, normal differences. But of course, all of that investigation itself had a cost. It caused anxiety in the patients and the participants. It caused lots of investigations, some of which could be harmful. And in fact, we know that even in that study, there were normal tissue removed. Normal ovaries were removed because something was seen on the ovary. And after investigation, it was thought better to remove it, and then it turned out to be normal. Bits of lung were removed, even though they then turned out to be normal. So what seems initially to be a good idea, that is, if you see anything, and you should really check to look for anything, it should be reported. It turns out that that can actually cause more harm than good. And so in the UK Biobank, if something is noticed during the imaging visit, then those few cases are checked and they are reported back. But I have to say, even then, we know that most of the time, those will actually not be things that matter. And much of the time, even the things that may be of concern, there isn't much that one would do about it. One would just perhaps watch it.
Thanks very much, Rory. So moving on to Steve, there's been a few questions about the pre-screening questions that we asked to make sure that you're safe to have an MRI scan. Now, you mentioned having a pacemaker, which would make you kind of ineligible for our scans. What are the other things that you think would be important for us to know? And in particular, if you have a hearing aid, does that mean you can attend one of the assessment centres?
Yeah, so as long as you're able to remove the hearing aid whilst you're in the scanner, then that should be fine. We do have quite a number of participants who have hearing aids. You'll be able to wear it throughout the rest of the visit, but when you're actually going into the MR scanner, you will be asked to remove it. If you're unable to remove it, if it is embedded, then you would likely be screened out. Other things to consider, if you've had metal in your eye, perhaps, and there is a risk that it is still there and actually in your eye, then you wouldn't, you'd likely be screened out. If you've had, there's different surgeries, but if you've had various implants or things in your abdomen especially, then you might be screened out. But the easiest thing to do is to give our contact centre a call when you receive an invitation, and they'll go through everything with you. If there's any doubt, then they'll refer that as well. So it's not just about safety either. There are some things that actually would be safe to scan, but actually they would make the images no use to us or the researchers. So we don't want you to come along to a visit if we can't actually use the subsequent images. So
great, thank you.
Okay.
And just could you comment on whether we provide travel expenses? I know some participants, particularly those living in Scotland, will have quite a long way to travel.
Yes, absolutely. There is a process for claiming your travel expenses, whether that's petrol for driving or if you want to come by train, then we'll refund your train fare, and there is a taxi service available from the main train station at Newcastle that will bring you to the site, and that's all covered.
Great, thank you. So Paul, I wondered if I could come on to you. There's been some questions in the chat about the research findings of the effect of coronavirus infection on the brain. And I wondered whether you could comment on how significant you think these findings are. For example, do you think they might be reversible?
So, Naomi, thanks. Those are really good questions. The significance, the question is whether they're reversible, whether they trigger a cascade of subsequent neurodegenerative changes of the type that we see more characteristically with dementias, or whether they're reversed, or whether they may just grow with time of their own accord. I think we don't know the answers to these questions at this point. The biobank, because of its design, allows us ways of answering them. For example, because blood was also collected on all of these participants, a study is underway, which we hope will produce results towards the end of the year or next year, which is looking at the relationship of the imaging changes to very sensitive measures of Alzheimer's disease and related neurodegenerative disorders in the blood. Actually probe whether these changes that are seen with imaging are associated with a cascade of other changes in neurodegeneration. One of the aspects of the disease, of the study that I probably didn't emphasize enough is that with the plan that Steve outlined to image as many of the 100,000 people again as is possible, we can expand the range of questions and actually ask whether these changes persist for a long time or grow in people who are imaged for the second time after longer delays to their initial exposure to SARS-CoV-2. The design of Biobank allows us to embed new kinds of case control studies over time as the questions arise. Another one that this would touch on, for example, I know was raised by someone in the audience about whether there's an effect of vaccination independent of exposure to the virus on the brain. I think the third thing, the final thing I want to say is that the importance of this resource, which I tried to stress, is that it's available for all researchers to work on together. And just as we saw in the pandemic, that's enabling a new kind of science in which everyone picks up the baton from each other and runs farther, answers more questions, and gets greater insight. I hope we've illustrated that today with regard to alcohol with the rather beautiful studies that Anya showed, which took the initial observations and is moving them to a new level and helping us to really assign risk on an individual basis even more precisely.
Thank you. In the interest of time, the final question goes to Anya, and there's been some comments in the chat about the interesting results of alcohol on the brain, with some comments on whether you think the brain can recover, and in particular, how useful might you think the having repeat imaging scans on participants will help you to answer that question?
Yeah, great questions there. Thank you. The short answer is I don't know. I think it's plausible that there is recovery because there are some small studies in very heavy drinkers that show some recovery on the brain scans is possible if people stop drinking. There aren't any studies that I'm aware of in moderate drinkers that have asked this question. Now, of course, the beauty of having the second scan at a later date is that we can start to try and look at these questions. And the other advantage, of course, is, some people say, okay, well, yes, the people drinking more have smaller brains, but how do you know people with smaller brains don't just drink more? And so having that second brain scan at a later point can really help tease apart those two things.
Wonderful, thanks. So maybe not all doom and gloom then.
Yes, we hope not, all of us.
So I'd just like to end by saying that after this event, all of you will be receiving an email from us in the next couple of days to invite you along to an imaging assessment centre where you can register your interest by clicking on a link in that email, and then someone from UK Biobank from our Participant Resource Centre will give you a ring to go through those screening questions that we talked about earlier to see if you're eligible to come along. There are 3 quick questions that we'd like you to answer before you go about how useful you thought this event is. And I'd just like to end by saying thank you to all of you who've committed your time and energy and continue to do so to help us to enable scientific discoveries that really do improve human health. So I hope you have found this informative, and hopefully we look forward to seeing some of you soon in Newcastle Imaging Assessment Centre. So thank you very much for attending and hope to see you soon. Thank you.